Howdy, hey, hi, and hello everyone, and welcome to Organelles. So, as you can see, we have this fully constructed eukaryotic cell right here. So let's go ahead and take out the organelles, and let's learn a little bit about each of them as we put this back together. So, that's what it looks like when it's all empty. Kind of funny, right? So let's start with the basics. So I've said the word eukaryotic quite a bit now, but what makes this cell eukaryotic? Well, there's two types of cells. There's a prokaryotic cell and there's a eukaryotic cell. And the way to remember which is which is this key little organelle right here called the nucleus. A eukaryotic cell will have a nucleus. However, a prokaryotic cell will not have a nucleus. So think of it like pro, no, and then eukaryotic, yes. That's how I've always remembered it. So, let's talk about the nucleus. This is what stores the DNA in your cells. So whereas a prokaryotic cell, which doesn't have a nucleus, the DNA would just be floating around the cell or contained in other areas, but it's not going to be concentrated like it is in the eukaryotic cell in this nucleus. So the RNA can go in and out of here from the ribosomes to the nucleus, and we'll talk about what the ribosomes do for this later. So this is the storage of DNA and the control center for the cell. So it tells things where to go. It tells that DNA and RNA where it needs to be. Now, before we put our nucleus back in our eukaryotic cell half here, let's talk about some of the other stuff that's inside of this part of the cell, this half here. As we can see, we have our cell membrane on the outside, but what's all this yellow stuff floating around? What are these little orange sticks we got poking out? What are the little red dots everywhere? Let's talk about that. So we're gonna go ahead and we're gonna start with these little orange sticks inside of the cell. So just like your cells have a skeleton called a cytoskeleton. You're gonna hear that part of the word cyte quite a bit in this series. And the word cyte just means cell. So we have cytoskeleton, the cell skeleton. So what does the cytoskeleton do? Well, the cytoskeleton, just as yours, provides a structure inside of the cell. It's a network of fibers that goes throughout the cytoplasm, kind of like the blood of the cell. It's this it's like a jelly-ish substance. This provides structural and mechanical support for your cell. It anchors organelles, which is why you'll see all of my cells, all of my organelles in this cell here have a little piece of cytoskeleton attached to them. That's what keeps them in place, if you will. This allows for vesicle and organelles movements as well as anchoring. So this occurs when the cytoskeleton interacts with motor proteins, which are just going to be proteins that move things, motor proteins. That's what's going to move the organelles around, say during mitosis or meiosis. So now that we've covered the little orange sticks in here, let's go over what these little red dots are going to be ribosomes. So why are there some in the cell and why are there some on this other organelle? Well, they have different functions, but the baseline is that ribosomes are protein synthesizers. Synthesis meaning to create. So these ribosomes that are just floating around inside of the cytoplasm, or cytosol as we might hear, these ribosomes are going to be the proteins that are produced out of these ribosomes are going to generally function within the cytosol, and they won't go anywhere else, and they'll function as enzymes. However, on this other organelle that we'll cover later, the proteins created from the ribosomes on this rough endoplasmic reticulum can be secreted through vesicles and be moved throughout the cell where they're needed. So if they need these ribosomes in the nucleus, if they need them to move other things, they can function as different. They'll be made to go where they need to be, essentially. Let's see here. What do we have? Oh. And now that we have covered the rest of what we can see in here, we'll go ahead, and since we've covered our nucleus, we'll go ahead and we'll put that right back in place there and keep moving. So next we are going to have the mitochondria So I think the mitochondria is probably one of just about everybody's favorite organelle, because the mitochondria is in fact the powerhouse of the cell. So this is going to be the site of cellular respiration. That's what it means to be the powerhouse of the cell. The mitochondria is not the only, but it is the main producer of ATP within the cell, which is the cell's form of energy, how the cell gets its energy to create these proteins with the ribosomes, and for the nucleus to be able to be the control center of everything. The structure of the mitochondria is actually pretty special because it has a double membrane. So this outer membrane here is going to be smooth, and the inner membrane is going to be made of folds called cristae. So the intermembrane is space between these two membranes, and the mitochondrial matrix, which is another fancy way of just internal cytoplasm-ish of the mitochondria, is going to be the location of the Krebs cycle, which is another fancy way of being part of the ATP creation team. And this is going to contain enzymes for catalyzing cellular respiration and ATP production, as well as mitochondrial DNA. It'll house that because the mitochondria has its own DNA. And of course, ribosomes, because where are they not? And the next organelle that we're going to pick up is this little purple one here that I've colored purple. I hope it goes without saying that the organelles are not actually these colors. But this one that I've color-coded purple here, this is going to be our Golgi complex. Some people have probably heard it referred to as the Golgi apparatus, but in our case, we've referred to it as the Golgi complex, so that's what term I am going to use. It is the FedEx of the cell. So the mitochondria is the powerhouse of the cell. The Golgi complex is the FedEx of the cell. This is what gets parts where they need to go. It contains flattened membranous sacs called cristae. We'll figure it out. These are separate sacs from the cytosol, and each cristae is not connected, so it's very difficult to display here on my little model here, but it's going to be like stacks together. They look like a little Wi-Fi symbol, if you will. The bars aren't touching there, but they're still pretty connected. And most importantly, this organelle has directionality. So for the nucleus, it doesn't matter which way it's facing, all different parts are gonna have the same thing. However, this other side here, it has a cis face and a trans face. Cis face is spelled CIS, and trans face TRANS. So the cis face is going to receive the vesicles from the endoplasmic reticulum. So vesicles, just the transporters. And the trans face is going to send the vesicles back out to other locations or to the membrane for secretion. Secretion is just going to be exiting, so it'll send stuff out through the membrane, out through the cell, to go to other locations. And obviously the functions, beyond being the FedEx of the cell, this receives transport vesicles with materials from endoplasmic reticulum. It modifies the materials, so if your package has a leak or something, I'm sure FedEx will probably just say, don't send it. But in our case, the Golgi complex is going to change that around and be able to send that out to where it needs to go. It's going to sort the materials that it's been given. So just as FedEx has its own sorting system, just like mail has its own sorting system, it's going to sort them so that way they can be better used for what they're needed. If the cell needs repairs, if it needs to start splitting, getting prepared for mitosis, then it can do that. And it adds molecular tags. So your mail has tracking numbers, adds little molecular tags on there. And it packages those materials to a new transport vesicle that exit the membrane via exocytosis. I'll go ahead and add this back in here. Does that look right? We'll find out. I'll move it around if I need to. Our next organelle is going to be the vacuole. This is going to be the storage of the cell. So in a plant cell, there's going to be one central vacuole that's going to be really big, filled with water. But in our animal cell case, they're going to be smaller, and there's going to be multiple of them throughout the cell. And there's types of vacuoles as well. There's a food vacuole, which sounds self-explanatory. It forms via phagocytosis, or cell eating, and then are digested by the lysosomes, which is the very next organelle that we will cover. There's a contractile vacuole, which is going to maintain the water level in a cell. And then there is a central vacuole, which, as I said, is going to be found in plants, and it's going to maintain the turgor pressure. So when your plant starts to wilt, it's because there's not enough water in the cells, in those plant cells, for them to maintain that turgor pressure, or internal pressure, to keep that plant upright. So it's going to start to wilt. That's why plants do that, which is really cool. Now that we have covered our vacuole, the storage organelle, we're going to go ahead and we're going to stick it right back in here. We're going to go with right there for now, and we'll move it around if we need to. As promised, our next organelle is going to be the lysosome, sometimes known as the trash can of the cell. And this is a membranous sac filled with hydrolytic enzymes. So enzymes eat, usually. That's a good way of putting it, is they do work. The function of this is to hydrolyze macromolecules in animal cells. Macromolecules are going to be your things like proteins, lipids, nucleic acids, as well as carbohydrates. That's what it is. The lysosome has this neat feature, which is referred to as autophagy. I'll correct myself if I'm wrong later. Which is how lysosomes can recycle their own material. So it's similar to like it's eating itself and reusing that stuff that it consumed, just reforming it to renew itself. So hydrolytic, that breaks down. What does hydrolytic mean? It means hydro, water. It breaks down things using water. So that's what that means. And then auto is self, phagy eat, so autophagy, it eats itself. So let's give it up for the lysosome. And as you might hear, Lysol, the cleaning product. So it's gonna be kind of cleans up after itself to reuse, renew, recycle. So let's add this back in here. I think I got that one right. I might be on a roll here. We'll see. And last but certainly not least, the one that we've been continuously mentioning but not bringing up is the endoplasmic reticulum. So there are two types. There's rough and smooth. And as you can kind of guess based on the red dots, the rough endoplasmic reticulum will have ribosomes on it, and the smooth will not. So these are a network of membranous sacs and tubes, and the functions are to synthesize membranes and compartmentalize to keep all of the proteins formed in the rough ER separate from those and those free ribosomes in our cell. And the two types. The specific function of the rough ER, and the specific thing that makes it fancy, makes it different, is that it contains the ribosomes that are bound to its membrane, so these aren't going anywhere. And the smooth endoplasmic reticulum, as we can see, has no ribosomes on it anywhere. And so this is going to synthesize lipids, and it's going to metabolize carbohydrates and detoxifies the cell. So detoxification is very helpful. Alrighty, let's put these right back in where they belong. I see a spot for this one right here, and I see another spot for our smooth right here. I don't think this is exactly how we started with this, but it still functions the same. So thank you for joining me to talk about the organelles of the cell. So what comes after the cell? Well, tissues. So stay tuned for the next video, and we will start our trip through tissues.